make that. I'll have to mix water, I'll have to mix agar-agar, and I have to take some more nutrient powder, I'll add it, and I'll make a nice paste, jelly-like paste out of it. And I'll pour it in a conical flask. Can you see the conical flask? There is some liquid jaisa material. That's jelly actually. That's not liquid, that's jelly-like material. ठीक है? So soil का replacement हुआ कि नहीं हुआ ये? Normal consideration में, normal criteria में it was soil. But when I'm doing micro propagation, I have to have this as a nutrient medium. Okay? What I will do now? I will take that explant, I will treat it with sterilization chemical, and I will put it in the nutrient medium. किसको ये समझ आ गया?
मुझे ज़्यादा नहीं।
अभी ये तुमको मैं बताऊं कि नहीं जानूं? I've taken an explant. What is explant? कोई भी एक शूटर मिक्स या बर्ड. उसको मैंने किस में डाला? स्टरलाइजेशन केमिकल्स में डाला और रख दिया साइड में. ओके? नेक्स्ट मैंने क्या बनाया? वाटर के साथ कुछ जेली जैसा सब्सटेंस and I've poured it in a conical flask. The next step is taking that explant and putting it in the conical flask. Is this easy now? Okay. Now can you see that this was the explant, right? But can you see it has started growing now, and it has grown to a little ball-like structure. ऐसा कुछ छोटा-छोटा leaf जैसा. This stage we call it as callus. क्या बोलेंगे? Callus. Can I say explant से callus बन गया? Easy. And when this explant that is made into callus, this callus will grow into a little plantlet. छोटा सा plantlet तुमको दिखेगा. Miraculous ना? Soil में उगना चाहिए था, इसमें हो गया. You'll see little plantlet. Now when this little plantlet comes out, you will take it from there and you will start putting it into the soil after some time. See?
अगर
हाँ? Is nutrient medium. पर वो दिया नहीं है तो लिखना होगा. समझ आया? Now why is this called as micro propagation? Because do you understand? I'm taking very little part of tissue and I'm growing it into a number of plant copies. और टिश्यू कल्चर भी हम इसीलिए बुलाते हैं because we are taking a little part of tissue. Easy? समझ आया? Now when it grows, it will be transferred. ऐसा लिखा होगा. The plantlets will be after some 4 weeks or 6 weeks. It will be transferred to the soil in a controlled environment. ऐसे नहीं कि कहीं भी उसको डाल दिया कुछ भी. Laboratory में हम अच्छा सा sterilized soil रखेंगे, उसको plant करेंगे. Then again have so many identical plants. Easy. Okay. अब टिश्यू कल्चर अगर तुमने सीखा तो तुमको ये भी अच्छा. There are 3 examples. 2 examples given to you. One is orchids, the other is gladiolus, and then we have chrysanthemum. ऑर्किड से पता होगा. ऑर्किड फ्लावर देखा हुआ है, पर्पल व्हाइट कलर का, शादी में लगता है इतना सारा फ्लावर. ग्लैडियोलस इज आल्सो वेरी गुड फ्लावर. क्रिसैंथेमम में इतना बड़ा सा फ्लावर होता है. ओके, सो ये बेसिकली ये प्लांट्स जो है हम उसको टिश्यू कल्चर के द्वारा वी कैन ग्रो इट. इजी. नाउ बिकॉज़ वी हैव स्टडीड दिस वी शुड नो द बेनिफिट्स ऑफ द सेम. बेनिफिट्स में क्या बोला जाता है? इट प्रोवाइड्स रैपिड प्रोपगेशन ऑफ आइडेंटिकल इंडिविजुअल. If I have very short period of time and I am expert in this, I can grow a lot and lots of plants, but they will be identical. Next, this technique is very productive for the superior varieties. Superior varieties का मतलब good varieties, good disease resistant varieties. अगर मुझे बना दिया, I can use this. Next is, it is very useful in cases in which the seeds are dormant and the embryo in these seeds can be cultured and micropropagated. What do you mean by this? Dormant का मतलब होता है, see, seeds will need 3 things. Normal seed will need 3 things. What 3 things? Water, good temperature and oxygen. जब तक सीड को ये तीनों चीज नहीं मिलता, it stays dormant. Dormant का मतलब inactive. That does not mean it's dead. I just said inactive. So it will not grow until it gets 3. जैसे मैंने अगर बोला मुझे एक स्कूटी चाहिए और मुझे ये चाहिए और मुझे ये चाहिए, only then I leave from here. If you give 2 also to me out of 3, I will not give. So seed will require all these things, otherwise it remains dormant. Now imagine if a seed stays dormant only, तो हम plant कब उगाएंगे? So in such case, I can do micro propagation. It's an easy method for me. If the seed is remaining dormant, जाने दो seed को जाने दो, ये micro propagation करते हैं. Easy. Limitation देखो. Limitation पे first क्या लिखा हुआ है? It
requires a lot of scientific effort.
अभी मैंने बोला ना कि ये करना पड़ेगा, स्टरलाइजेशन केमिकल ना. If you are very new, and I tell you micro propagation करो, मैं जाके आ रही हूँ तब तक. You don't know ABCD of it. What are you going to do out of it? तुमको सब पता होना चाहिए कि which chemicals are needed, how to prepare the nutrient medium. मैंने बोला था ना. So if you don't know, you will do crap. क्योंकि उसमें है ना measure करके सब डालना पड़ता है. If you don't know the proportion, it will turn into solid. It will not have jelly. समझ आया? And next, what do we have? It cannot be applied to all cases. See, the example given to you are what these examples. I cannot do this with all the plants. I will not get the result. It will fail. So I should know that if I am doing macro propagation, what all options I have? क्या क्या उगा सकते हैं हम इस तरह से? And lastly, it cannot be easily applicable in remote areas. So if I don't have a lab setup, do you think this is possible? I have to have a lab and control setup. And we cannot expect this to be happening into rural areas. I cannot do micro propagation there. Okay, so I have to have a lab setup and all. इसीलिए वो disadvantage हो गया. Okay, we will watch a video so that you understand. And you know, I also did this when I was learning myself. When I was in college, we did micro propagation. और मैंने अभी क्या बोला था? इस plant में हम क्या लेते बेटा? Bud or shoot apex. Do you know what we do? And I want you to go home and check this. Cabbage देखा हुआ है? Cabbage कैसा होता है देखा है? Overlapping leaf ऐसा. Okay, go home today if you have cabbage, very well and good. If you don't have, tomorrow maybe day after tomorrow. धीरे से एक पत्ता उसका ऐसे निकालो नीचे तक, base तक. You understand? Just don't tear it like this. Okay, nicely take it out. And when you go towards the base, if you are very observant, base पे there is a little triangular structure that you will see. We do that as an experiment. I'll show you if you want. Okay, so that you understand. I can explain you this way, but it's not very well there. This is half of the cabbage. Okay. When you go home and peel one leaf properly onto this area, ये base है ना, onto this area, see you will find something like this. Can you see little portion white color ऐसे? Raja, तो please देखो पहले cabbage है. We took that little portion out. That's called as vegetative bud. We took that little portion out and then we did this whole thing. I'll show you a video of vegetative propagation so that you remember. Sorry, होता है, इसका दिख जाता है इसीलिए. Okay, otherwise then I'll have to take shoot apex for other plants. between yours
Tissue
and
culture.
don't break my monologue.
The technique of tissue culture has
Is it
emerged
okay?
as a very efficient tool to propagate better improved crops. Tissue culture is a much approachable technique than other traditional plant breeding techniques. By using this method, cells, tissues, etc. are maintained and grown in vitro under aseptic conditions in artificial medium, providing necessary nutrients for growth. In 1839, Schneider and Schwann proposed that cell is the basic unit of life and is capable of autonomy. Each cell in an environment is capable to regenerate into a whole plant. Based on this theory, a German physiologist, Gottlieb Herberland, in 1902, developed the concept of in vitro cell culture.
You don't have much of this, just study when the tissue culture comes.
Cells in his experiment increased in size, but failed to divide, due to which his predictions failed. However, his hypothesis paved the way for more research. He is thus regarded as the father of tissue culture, taking cue from his experiments, having in 1907 to nearly mature embryos of crucifers and successfully grew them to maturity. In 1908, Simon regenerated callus, buds, and roots from popular stem segments and established the basis for callus culture. A breakthrough came from White in 1934, who experimented on tomato roots. He reported not only could cultured tomato root tips grow, but they could be repeatedly subcultured to fresh medium or inorganic soils, supplemented with yeast extract.
Floriculture?
has been the
Floriculture is growing of flowers.
characteristic.
This agriculture is for crops, floriculture is for flower, and horticulture भी बोला था ना? Horticulture is for vegetables and fruits. Because if I am growing fruits, I cannot say agriculture.
Crops also are a part of agriculture that they grow vegetables.
So when I say, as a farmer, I say I'm into agriculture, I'm growing cereals more. What are cereals? Wheat, rice, bajra, jawar, all of this. But a specific term for growing of vegetable is horticulture, and for flower it is floriculture. Okay, so many times this is used for floriculture also. इसीलिए तुमको examples क्या दिए देखो, flowers ही दिए ना. For example, orchids, gladiolus, all of these are examples. Now I want you all to read this very carefully going home because I'm not. I think we can start with this. I'll just explain you, introduce you this. However, I'll take your test till year only, so that you are well prepared for your exam. Starting from 11th, na, your exams, and then bio is 18th something.
I think my biology is
Last.
first week only.
Okay. May, whatever it is, whenever 11th or whatever. So अभी तक हमने क्या किया? What we have done so far is vegetative propagation, artificial propagation, all of this, right? Now we are going to sexual reproduction in plants. अभी तक क्या था? Asexual reproduction. Now we are going to sexual reproduction in plants. So the sexual reproductive part of a plant is what? Flower. समझ आया? मैंने तुमको स्टार्टिंग में बोला था. I don't know if you remember. If at all you talk about plant, what are parts it has? Root, stem, leaf, flower. So flower को छोड़ के all these three are called as vegetative part, and flower is called as the reproductive part. Okay. Now this flower that you see, okay, this flower that you see will have 4 whorls into it. The flower is the reproductive part. Okay, it is written over here. And the flower is first of all having a stalk. Now I have just drawn a little portion of stalk, but I am sure you know about stalk. Rose देखा होगा? Rose में एक होता है ना ऐसे डंठल, the stalk-like portion. So this is stalk. The technical term is also called as pedicel, but I am not sure. It's given to you. Yeah. So if I write stalk, or I can write it as pedicel. It's already given. Easy. The next we have is, other than that, you have been given a cup-shaped structure. Can you see a cup-shaped structure here? Just above the stalk. Every flower will have this. This cup-shaped structure that you see is called as thalamus. Easy? ये क्या काम करता है? Stalk क्या काम करता है? It keeps a flower in upright position. The thalamus will hold all of this together. So it is thalamus. It is one cup-shaped structure that holds everything together. Next we go to the 4 parts of the flower. But हम parts नहीं बोलेंगे. अभी तो इतने छोटे से नहीं होते तो parts बोले. So there is a different word for it. We say whorls of flower. What do we say? Whorls of flower. Whorls is nothing but parts. Okay. So we have 4 whorls of the flower. The first one I will teach you is calyx. The second one will be corolla. The third part will be androecium. And the fourth part will be gynoecium. Easy. Very good. इसमें मार्क करेंगे, पर इसमें ये मार्क नहीं करूँगी. This is the whole group name. But the calyx is made up of what? It is made up of sepals. Have you seen sepals? Have you seen a rose plant? Rose flower. उसमें वो green color का नीचे के side होता है, rose तो red है. नीचे green color का छोटा छोटा nucleus structure, that is sepal. And if you have counted, you will understand there are 5 sepals. So एक होगा तो मैं sepal बोलूँगी. अगर मुझे पांचों एक साथ बोलना है तो मैं बोलूँगी calyx. Can I say calyx is made up of sepals? Can I say that way? The corolla, on the other hand, is made up of petals. Very good. Okay. The androecium is made up of stamen. Very good. The stamen right there will be made up of what? 2 things together. It has 2 parts, anther and filament. and the gynoecium will be made up of pistil. The pistil has three parts again.
Ovary, style,
Stigma, then style, and then ovary. And in ovary there will be ovule. Okay, let's mark it there very quickly, and you will have to remember this and draw this and come next time. Okay, thalamus is done. Can we mark sepal now? What is this? Green structure here and here, both of them are sepal. These large structures that you see, which are usually colorful, this will be marking as petals or petal. You see? Can you see this particular structure right here? This top portion of the structure is called as anther, and this thread-like structure is called as filament. Can I group them and say that this is a stamen? This is a, and can you see there is 1, 2, 3, 4 stamens? Together they will be called as endometrium. You see? Next up, the top knob-like portion will be called as the stigma. This structure, tubular structure connecting this and this portion will be called as style. And this basal portion that you see would be called as ovary. Can I say that these three together is called as a pistil? Easy? ये pistil मैंने एक ही draw किया है. What if I draw 2, 3? Then together all of them will be called as light. This is easy. And what is this?